Welcome to The Explainer. Now, if you or a loved one has ever had to navigate cancer treatments, you already know the side effects can be, well, absolutely brutal. Today, we're exploring what is arguably the holy grail of modern cancer research. See, for years, doctors have been using the incredible power of our own immune systems to fight cancer, but there's always been this massive catch, right? How do you unleash the immune system to ruthlessly attack a tumor without it turning around and just destroying your healthy tissues? Well, today, we're breaking down a groundbreaking new study that might just let us cure the cancer completely with the collateral damage.
Okay, let's dive into this. Here's our roadmap for today. We're going to start with the immunotherapy dilemma, then explore the paradigm shift, unmask CRTAM, the toxicity culprit, look at proving the concept, and finally, we'll ground ourselves with limitations and future steps.
Starting with Section 1, the immunotherapy dilemma and the double-edged sword of ICB.
Current treatments like immune checkpoint blockade, or ICB, they're absolute miracles for so many patients. Why? Because they essentially take the brakes off the immune system so it can hunt down and destroy cancer cells. But the medical world really struggles with what are known as immune-related adverse events, or IRAEs. And to be clear, we aren't just talking about a mild rash or a slight fever here. We are talking about literal friendly fire. Severe, life-threatening immune system attacks on normal, healthy tissues. This is the biological collateral damage happening in patients right now, because the immune system is basically doing its job a little too well, and it can't always distinguish between the enemy tumor and the patient's own healthy organs.
For the longest time in cancer research, there was this massive underlying assumption about all this. If you look at efficacy on one side, which is the tumor elimination, and toxicity on the other, which is the severe collateral damage, scientists largely assumed these two outcomes were just permanently bundled together. You know, a package deal. It was thought that if you wanted the immune system to fight the cancer hard, well, you just had to accept that it was going to fight the healthy tissue hard too. Moving to Section 2, the paradigm shift, and changing the fundamental question.
Instead of focusing broadly on how tumors evade the immune system, the researchers here decided to just flip the script. They changed the question entirely. They went hunting for immune molecules that are exclusively linked to toxicity. They asked this incredibly provocative question: what if we can actually decouple the cure from the collateral damage? Like, what if there's a specific pathway, a toxicity-specific immune checkpoint, that we can just block to stop the autoimmune damage without dialing back the immune system's attack on the cancer? Finding a molecule associated strictly with toxicity and not efficacy, that would change the world of oncology forever.
And they didn't just guess either. They executed a seriously rigorous strategy. Step 1, sifting through massive amounts of transcriptomic data and FDA patient records to identify toxicity molecules. Step 2, establishing causality in animal models. Then, mapping the exact tissue targets, defining the inflammatory mechanisms, testing that separation of toxicity and efficacy in tumor-bearing mice, and finally, exploring human clinical biomarkers. It's honestly a masterclass in comprehensive scientific methodology.
All right, Section 3, CRTAM, the toxicity culprit, finding the rogue molecule.
Out of all that massive mountain of data, one surface molecule emerged as the clear primary driver. It's called CRTAM. It was heavily associated with causing toxicity, but get this, it had absolutely no connection to treatment efficacy. To understand why CRTAM is so incredibly dangerous to healthy tissue, think of it like a rogue GPS system inside our immune's T cells. These T cells are circulating in the blood, actively looking for cancer, but when a T cell has CRTAM on its surface, it acts like this awful homing beacon that completely misdirects the T cell away from the tumor and straight into healthy epithelial tissues. Once it's there, it binds to a specific partner molecule called CADE-M1, and boom, this interaction ignites a massive localized type 3 immune fire.
And this brilliantly illustrates exactly how this biological chain reaction plays out. You've got CRTM on the T cells acting as that rogue GPS, and it's actively searching for CAD-M1, which is highly expressed on normal epithelial tissue, the destination. When these two finally connect, they trigger what's known as the IL-23 and IL-17 inflammatory axis. This perfectly explains where the toxic immune response happens and exactly why healthy tissue gets destroyed, while the tumor is sometimes just perfectly left alone by these specific cells.
Bringing us to Section 4, proving the concept from mice to humans.
To prove this causally, they didn't just look at one random organ. I really want to highlight the number 3 here, because one of the absolute greatest strengths of the study is its cross-organ relevance. By genetically deleting or blocking CRTAM, the researchers successfully protected 3 major organ systems: the colon from colitis,
the lungs from pneumonitis, and the heart from myocarditis. This strongly suggests that this CRTAM pathway isn't just a one-off. It could represent a broad, universal mechanism for immunotherapy toxicity.
But here is the absolute massive win, the true holy grail moment to the whole study. When they knocked out CRTAM in tumor-daring mice, they saw drastically reduced colitis and pneumonitis. But crucially, and this is the amazing part, they maintained tumor control and they preserved the tumor-infiltrating T cells. By taking out CRTAM, they cured the collateral damage without ruining the immune system's attack on the cancer. They literally separated efficacy from toxicity.
Now, what's really interesting about these results is how they actually translate to humans. The researchers looked at independent patient cohorts and mapped their baseline CRTAM expression against the incidence and severity of adverse events. The data is just incredibly clear. Patients with high baseline CRTAM experienced much more frequent and much earlier severe toxicities, which means CRTAM isn't just a therapeutic target, it's a potential predictive biomarker, one that could actually tell doctors who is at risk before treatment even begins.
Finally, Section 5. Limitations and future steps, the reality check. Look, as wildly exciting as this is, science takes time. Jumping from preclinical success to everyday clinical use is a massive, massive leap. We have to starkly contrast what is definitively proven versus what is still unproven. The researchers have absolutely proven preclinical success in mice, and they've proven human biomarker associations in observational datasets. But mouse models, like chemically induced colitis, they're simplified. They just don't perfectly replicate the complex, messy nature of human adverse events, which are constantly influenced by genetics, your microbiome, prior therapies. So what remains entirely unproven is prospective clinical efficacy and long-term safety in humans.
So what are the crucial next steps? Well, first, randomized human clinical trials are an absolute must. We need to validate if testing for CRTAM and IL-23 actually predicts risk in the real world. We also have to figure out timing. Do you block it prophylactically before treatment even starts, or only reactively after toxicity appears? And most importantly, safety. We have to make absolutely sure that turning off CRTAM doesn't accidentally compromise a patient's long-term immune memory, or, their ability to fight off regular everyday infections. So there is definitely a lot of vital work ahead.
But the foundation laid here is nothing short of revolutionary, which really leaves us with this final thrilling question. What does the future of cancer treatment look like now that we know we might actually decouple the cure from the collateral damage? We are looking at a true paradigm shift in oncology here, folks. A future where treatments can be both fiercely effective and fundamentally safe. I don't know about you, but I'm incredibly energized to follow the future of this CRTAM pathway. Thank you so much for joining me for this explainer. Keep questioning, keep learning, and I'll see you next time.